Good morning. How are you? Today, inshallah, we will complete with chapter 12, the DNA. Listen to the structure of the DNA. So, for my objective today, to study the DNA structure. What we need to know about the DNA structure today, the main structure, the component, the chemical formula for each of the components of the DNA structure. So, as we learned last lecture, we know that the DNA, which is the genetic material. The main component for the DNA, which is the nucleic acids, are long, slightly acidic molecules originally identified in the cell nuclei. So where we can find the DNA or the nucleic acids, you have to know this information. The nucleic acids is similar to the DNA, is the DNA. The nucleotide is the component of the DNA. So the DNA is found inside the nucleus. Nucleic acids are made up of nucleotides. The nucleotide is the monomer for the nucleic acids, linked together to form long chains. So nucleotide is monomer for nucleic acids. DNA nucleotides are made up of three basic components. So the main idea, you have to know the basic component for the DNA, for the nucleotide, which is 5-carbon sugar called deoxyribose, a phosphate group, and nitrogenous base, and we have 4 nitrogenous bases. So again, the DNA, which is abbreviated for deoxyribonucleic acid. So DNA, deoxyribonucleic acid. Deoxy related to deoxyribose, which is the 5-carbon sugar without and 3' oxygen, phosphate group, and nitrogenous bases. As we mentioned before, the nitrogenous bases that is found in the DNA is 4 nitrogenous bases, which is adenine, guanine, cytosine, thymine. Again, adenine, guanine, cytosine, thymine. As you can see here, adenine and guanine known to be 2-ring cycle, and cytosine and thymine 1-ring molecule. And all of the bases, nitrogenous bases, are attached to deoxyribose, and the phosphate group is attached to the sugar, deoxyribose sugar. Okay? Again, the nucleotides in the strands of the DNA are joined by bond. This bond is known to be covalent bond, formed between their sugar and phosphate group. So the bond between this nucleotide and this nucleotide is between the sugar and the phosphate group, between the sugar and phosphate group. This is the bond. This bond is known to be covalent bond. Again, the bonds between this nucleotide and this nucleotide, it's between the sugar here and the phosphate here, and the bond is covalent bond. Okay? Again, the DNA is made up of 4 nucleotides, A, G, C, T. A for adenine, G for guanine, C for cytosine, and T for thymine. You have to memorize this information. The first one who tried to understand the structure of the DNA, which is Giraffe or Erwin Giraffe, he discovered that the percentage of adenine and the thymine bases are almost equal in any DNA sample. At the same time, he discovered that the guanine and cytosine also have the same percentage. So he observed that the A always bound to T, while C always bind to G. So again, T is bounded to A and C is bounded to G. So A always bound with 2 bonds with T, while C always bound with a 3 bond with G. You have to know that. These bonds known to be hydrogen bonds. Again, this is a hydrogen bond, okay? Between the 2 nucleotides, between the bases, 2 bases, is hydrogen bond. Always A binded to T, while C always binded to G. Okay? Understood? Okay. Another scientist who has already studied the structure, which is Rosalind Franklin, which she's used the X-ray diffraction to reveal an X-shaped pattern that showed already under the UV. This is an, X-ray. This is an X-ray pattern for the DNA, and this pattern shows that there is a helical shape. So the strands of the DNA are twisted around each other like the coils of the spring. So, like this, okay? So that there are two strands in the DNA, the observation of X-ray that's done by Franklin, that there is 2D strand of the DNA structure, and there is a nitrogen bases maybe near to the center. So in the center we have the nitrogen bases, and there is 2 strands of the DNA molecule. The work were done by Franklin is used by another scientist, which is Watson and Crick. Watson and Crick used the picture obtained by Franklin X-ray, and they explain the specific structure and properties of the DNA. And by using the X-ray pattern, they built 3-dimensional model of DNA in a double helix, in which 2 strands were wound around each other. So Watson and Crick used the structure or the X-ray pattern obtained by Franklin, and they form a 3-dimensional model of the DNA. After they already built a 3D structure of the DNA, they found that the DNA strands anti-parallel strands. What do you mean by anti-parallel strand? So, anti-parallel, which is meaning they run in opposite direction. If we say this is the direction of the first strand from 5 to 3, the other direction from 5 to 3 in the opposite direction. So, again, this is the direction of the first one. The direction of the other one is like this. So if we started here with phosphate, we will start here with phosphate. If we end here by sugar, ribose sugar, deoxyribose sugar, here we will end with deoxyribose sugar. Okay? This is what we mean by anti-parallel, which is running in opposite direction. This arrangement enables the nitrogenous bases on both strands to come into contact at the center of the molecule. So in the center, we have the nitrogenous bases, and we have on the backbone, which is phosphate sugar backbone. So this is a phosphate sugar backbone, and in the middle we have the nitrogen bases. And the nitrogen bases, as we mentioned before, are attached by the hydrogen bonds, okay? So again, the DNA is made up of 2 strands. These 2 strands are known to be double helix in a helical shape, and the strands are made up of a phosphate sugar backbone, and in the center we have the nitrogenous bases attached to each other by hydrogen bonds, and at the same time the 2 DNA strands are anti-parallel strands. These information are very important about the DNA structure. You have to know that. Again, the direction, it's from 5 to 3, and at the same time both of the strands are anti-parallel to each other, and in the middle we have the nitrogenous bases with the hydrogen bond. The hydrogen bonding, what do you mean by the hydrogen bonding? The hydrogen bonding is the bonds between the nitrogenous bases. As we mentioned before, the C and G nitrogenous bases are attached by 3 hydrogen bonds, while the A and T are attached with 2 hydrogen bonds. For the hydrogen bonds, it are relatively weak chemical forces that allow the 2 strands of the helix to separate. So it's a good way to attach with the hydrogen bonds. Once the DNA needs to duplicate, can separate and duplicate and then reform again. So the ability of the 2 strands to separate is critical to the DNA function. If the DNA strands can't separate, this is really hard for the cell to duplicate or replicate the DNA. Again, for the base pairing, we say that the G attached to the C and T attached to the A. You have to memorize all of these things as a base pairing. And till now, we complete our lecture. Thank you and have a nice day. Inshallah, today we finished chapter 12.2, and tomorrow, inshallah, we will complete with the next lesson.